Okay, so welcome back to this next video in which we are discussing the cell cycle. Okay, right, so where we left off was we were at the end of the G2 phase and we were now going to move into M phase. Okay, so let me just remind you of where we have got to. We now have an enormous great cell, okay, that has enough intracellular organelles and enough protein for 2 cells. Then it has a nucleus which has 2 copies of every chromosome. Okay, so here I'll draw one chromosome that's got 2 copies of itself here, which are attached together via cohesins, and these are called the 2 sister chromatids. Okay, so we have 46 sister chromatid pairs, basically. Okay, right. So that's where we've got to at the end of G2 phase. We are now ready to move into M phase, in which we are going to perform mitosis, which is the nuclear division. Okay, so we're going to split the nucleus in 2, and we'll go from having one cell with a single nucleus which has twice the copy of DNA to having one giant cell with 2 nuclei with single copies of each chromosome. Okay, and then we'll go through the process of cytokinesis, which is the process whereby we split this massive cell with 2 nuclei into 2 cells with 1 nuclei. Okay, or 1 nucleus rather. Okay, so what then is going to activate the movement from G2 to M? Well, basically, at the transition between the second gap phase and the mitotic phase, what happens is that the level of a protein called CDC25 goes up within the cell, and it is not known what causes this to happen, okay, but this is extremely important that this protein goes up, because this protein is a phosphatase enzyme. It removes phosphate groups from proteins, basically. Okay, so CDC25 is a phosphatase. I'll just write this down. Okay, and what it's going to do is when it goes up in the cytoplasm of the cell, it's going to now remove those phosphate groups that We1 has put onto the tyrosine and threonine residues that are in the active site of the cyclin-dependent kinase 1 enzyme that is in a complex with cyclin B. Okay, so what you're going to get, therefore, is when CDC25 goes up, you're going to get the activation of the cyclin B CDK1 complexes. Okay, so let me draw cyclin B here. So this is cyclin B in blue here. Okay, and it's bound to cyclin-dependent kinase 1. Okay, so here is cyclin-dependent kinase 1. And cyclin-dependent kinase 1 previously had these 2 phosphate groups on tyrosine 15 and threonine 14 that were within its active site. Okay, but now, thanks to the CDC25 phosphatase, these phosphate groups have been removed. However, the phosphate group that is attached onto threonine 161, okay, so this is threonine 161 here. Remember, the single letter code for threonine is T. And this has been attached via the cyclin-dependent kinase activating kinase, CAC. This is not going to be removed by the CDC25 phosphatase. Okay, so CDC25 just removes those phosphate groups that are inactivating the cyclin B CDK1 complex. So now we have this cyclin B CDK1 complex that has this activating phosphate group on it, and therefore this is going to become active now. Okay, so we're going to start getting the activation of these cyclin B CDK1 complexes. Now, the next thing I want to talk about is the fact that there is a positive feedback loop with regards to the activation of cyclin B CDK1 complexes, because once this complex is activated, it's going to phosphorylate CDC25 enzymes. Okay, so here it comes. It will phosphorylate the CDC25 enzyme, like so. And this is then going to activate CDC25 further. So now CDC25 will gain even more activity, and it will go off and activate loads more of the cyclin B CDK1 complexes that at the moment are sitting in the inactive form because they have phosphate groups attached to tyrosine 15 and threonine 14 within their active site. Okay, so remember, you have a massive, great pool of these inactive cyclin B CDK1 complexes that are built up in the cytoplasm of the cell throughout G2 phase. Okay, at the beginning of M phase, you're going to get the sudden activation of CDC25. Okay, and this will start by converting a few of these complexes into the active cyclin B CDK1 complexes. But then we're going to get a positive feedback on this loop because the cyclin B CDK1 complexes that have been activated by the CDC25 will now phosphorylate the CDC25, and they will lead to CDC25 becoming more active. So the rate at which it activates further cyclin B CDK1 complexes that are currently in this inactivated state because they have phosphate groups on tyrosine 15 and threonine 14 is going to get greater. So you're going to get more cyclin B CDK1 complexes that are in the active state with no phosphate groups in their active site being formed. In addition, there's a further way that there is positive feedback because the cyclin B CDK1 complexes are going to phosphorylate and inhibit. That's what a line with a perpendicular edge like that means, inhibits. Okay, they're going to phosphorylate and inhibit WEE1 enzymes. Okay, so they're going to inhibit WEE1 so that WEE1 can no longer rephosphorylate the cyclin B CDK1 complexes that are active and thereby inhibit them. Okay, so both of these things are going to help turn this pool of cyclin B CDK1 complexes that are in this inactive state into loads of cyclin B CDK1 complexes that are in the active state. So basically, what's suddenly going to happen now is all of the cyclin B CDK1 complexes that we had, which were sitting inactive because they have phosphate groups on tyrosine 15 and threonine 14, are suddenly going to be turned on into this on state where they no longer have those phosphate groups bound to tyrosine 15 and threonine 14. Okay, right, so that's the turning on of the cyclin B CDK1 complexes, which remember are also known as MCDK. Okay, the activation of MCDK then is going to guide most of the first phases of mitosis. Okay, so I now want to discuss the phases of mitosis with you, and I'll point out where there is a process that is dependent on the MCDK enzyme, okay, or the cyclin B CDK1 complexes. So, mitosis then, once again I will remind you that mitosis, strictly speaking, does not mean the division of the cell. It means nuclear division, not the division of the cell. Cytokinesis is then cytoplasmic division. Okay, right, so the first stage of mitosis, and I'll redraw this picture of the cell that we currently have. Okay, so currently we have a very large cell with a nucleus that contains 2 copies of every single chromosome. So all 46 chromosomes, we have 2 copies of them, here and here. Okay, so for simplicity, all the way through, we'll just show 1 chromosome, but the principle is the same when you've got 46 of the things, okay? In fact, actually, maybe I'll show 2. It might be a little bit difficult to show metaphase with only 1 chromosome, but so I'll show 2. So there we go, we have 2 chromosomes, okay? And each of these chromosomes, we have 2 copies of them. Okay, so overall we have 4 chromosomes, if you like, there, but obviously they're 2 pairs, really, 2 pairs of sister chromatids. Okay, right, so the first phase then of mitosis is what's known as prophase, and the 2 things which occur in prophase are completely orchestrated by these cyclin B CDK1 complexes. So the first is we're going to get what's known as chromosomal condensation. And we're also going to get what's called chromosomal resolution. So we're going to get chromosomal condensation and resolution. So what does this mean? Well, basically, prior to the prophase of mitosis, the sister chromatids that we had within the nucleus were not all aggregated together into a visible structure. Instead, this picture's misleading, basically. Instead, what the nucleus would actually look like is something like this. Okay? So the chromosomes are just all over the place, basically. They are just huge, huge pieces of DNA, and they are not condensed at all. They're just all over the place, basically. It's just a meshwork of pieces of DNA all over the place. Okay? And when the DNA is in this form, it's known as chromatin. So, at the moment, you do have all of the chromosomes having been copied, okay? So all of the pieces of DNA that are in here, all of the double-stranded pieces of DNA, they are sitting alongside an utterly identical piece of DNA, held together, remember, by the cohesins that we've discussed. However, at the moment, they are not condensed into visible structures. They're just all over the place. Okay, so what happens as one of the first phases of mitosis in this prophase is that the nucleus will go from looking like this to looking like what I've drawn here. I drew it like this because obviously this, you can't see anything, okay? I drew it like that because it's easier to understand that, but in reality it will look more like this, okay? So what happens in the first phase of mitosis, in prophase, is that the chromosomes do actually turn into something that looks like this, okay? So the chromosomes condense into visible structures, okay, and the two sister chromatids are still attached to one another. So here we have our two chromosomes, basically. Here are our two sister chromatids that are red, and here are two sister chromatids that are blue. So basically what happens is all of the DNA in one chromosome condenses down into a very packed structure which is actually visible, okay? And you have to understand that this is not going to be, this is not a linear piece of DNA now. This is not just the two strands in a linear line like this. Instead, it's hugely condensed up. You have wrapped the DNA around histones. You've packed those in very tightly together, okay, to make this visible rod structure here. Okay, right, so that process of condensing the DNA down into these large rod structures that are actually visible is known as condensation, okay, or chromosomal condensation in full. Now, in addition to chromosomal condensation, what's also going to happen is you're going to have chromosomal resolution. Now, what does that mean? Well, originally, when we had the two chromosomes, okay, that were identical to one another, and this is really confusing because this picture was supposed to be the double-stranded DNA, and this picture in red, these are supposed to be the chromosomes. So I'll draw it out in full. Okay, so here we now have our two double-stranded pieces of DNA. Okay, so to make this utterly plainly clear, I'll colour these two strands in red because these were the two strands of the original chromosome. And, oh no, now I'm going to confuse it with this. I need to use a different colour. Okay, so this is part of the confusion of this, that I'm using rods to show really dense DNA packed up, rods to show a chromosome that's linear, and then rods to show an individual strand of DNA, but never mind. Okay, so I've got the two pieces of DNA here. So this is a piece of double-stranded DNA here, okay? And this is a piece of double-stranded DNA here. And these two we are assuming are identical to one another. Okay? So originally these had cohesins around them all over the place, holding them together. Now, when you condense the chromosomes down to form these two rods that sit next to each other, the links between the two sister chromatids have to become much weaker. Okay, you can't do this basically unless you break apart some of these cohesins. Okay, so you hugely weaken the interactions between the two sister chromatids, and this breaking down of many of the cohesins is known as resolution. Okay, there will be a few cohesins. So if I draw this picture larger, often people will draw you a picture like this. Okay, so these are the two sister chromatids now. So this is one sister chromatid, and it's not a linear piece of DNA anymore. This is a huge, great rod with DNA stored within it. Okay, and then on the other hand, we have our other sister chromatid here in blue. Okay, so these are the two identical chromosomes condensed down. Now, there will be little points where you have, you know, a little bit of DNA stretching out maybe from here, a little bit of DNA stretching out from here, and then there'll be a cohesin around them. So there are cohesins holding these two sister chromatid structures together now. Okay, but the place where they're most tightly held together is this central bit here. Okay, and the central bit here is known as the centromere, or also as the kinetochore. Okay, and that's a name we're going to come back to, because this is the portion where the microtubules that are going to be involved in pulling the two sister chromatids apart are going to attach. Okay, so this is the centromere, and it's got the kinetochore within it. The kinetochore is the name for a special attachment structure that will attach onto the microtubules. So this is the centromere. Okay, right. So we're going to get chromosomal condensation and resolution. Condensation involves the DNA tightening up to make these macroscopic visual rod structures. Okay, resolution means that we're going to have to break apart many of the cohesins in order to form these visible rod-like structures, although we'll keep a few of them, which are holding the two sister chromatids still together. Okay, right, and all of this process of the formation of these two visible sister chromatids attached together mainly at the centromere here, okay, all of this is governed by the mCDK, okay, so by cyclin B CDK1 complexes. Right, so that's one of the things that's going to occur in prophase. The other thing that's going to occur in prophase is the assembly of the mitotic spindles. Okay, so what does this mean? Well, the mitotic spindle, rather, not the mitotic spindles, the mitotic spindle, is the structure which is going to pull the sister chromatids apart later on. Okay, and it consists of meshworks of microtubules that are stretching out from two structures known as centrosomes. Okay, so in this dividing cell, you are going to have two of these structures which produce microtubules, and these are called centrosomes, and they are intracellular organelles. Usually a cell would only have one centrosome. Okay, but in the division process, what happens is you get centrosomal division, and therefore by this phase of the cell cycle, you've got two centrosomes. Okay, so these centrosomes will start to produce microtubules that start to stretch towards one another like so. And this entire structure, the two centrosomes with their microtubules that are stretching out, the whole structure is known as the mitotic spindle. It's the structure which is going to pull the two sister chromatids apart. So here are the centrosomes in turquoise, and then stretching from the centrosomes, we then have the microtubules in pink here. Okay, right, and that entire structure then is called the mitotic spindle. Okay, right, so that's prophase. You get chromosomal condensation in the nucleus, and you start to form the mitotic spindle in the cytoplasm. And both those processes are governed by this mCDK, cyclin B CDK1 complexes. Okay, right, so the next phase of mitosis is what's known as prometaphase. Okay, and in prometaphase, what's going to happen is the nuclear envelope is going to break down. Okay, so the membrane that surrounds the nucleus is going to break down, releasing all of the now condensed chromosomes into the cytoplasm. Okay, so let's draw this. So here we have our cell, like so, and now the nuclear envelope is gone. So we've now got our two sister chromatids, which I'll draw like so, and these are in the cytoplasm now, and I said we were going to draw two chromosomes, so I'll keep to that. So we'll have chromosome in red here, and these are the two sister chromatids now held together at the centromere, but of course you have cohesins all the way along. The centromere is the point where the two sister chromatids are closest together. It's also the point where you have this kinetochore, which is going to be involved in binding to the microtubules of the mitotic spindle. Okay, then we also have chromosome blue here. Okay, so nuclear envelope has gone, basically. This process of the nuclear envelope breakdown, this is again governed by the MCDK. Okay, so it's responsible for the nuclear envelope breaking down. So MCDK is pretty much responsible for all of the first portion of mitosis. Okay, right. What then is going to also happen in prometaphase is you're going to get chromosomal capture. So let's say that these two things here, which I'll colour now in turquoise, these are the two centrosomes. Okay, so here is one centrosome, and there over there is the other centrosome. Okay, right. So from these centrosomes you have many microtubules coming off, and now what's going to happen is a microtubule from this centrosome over here will attach to the kinetochore of this red sister chromatid pair here, okay, and it will attach to the kinetochore on the side of this red sister chromatid to the left, and then the other red sister chromatid will be attached to a microtubule from the other centrosome, and it will be the same for the blue sister chromatids here. So these two centrosomes, they are known as the opposite poles of the mitotic spindle. So you have two poles of the mitotic spindle, one pole over here and another pole over here. Okay, and basically each pole is going to get one of the sister chromatids within the sister chromatid pairs. So one sister chromatid is going to attach to a microtubule from this pole, and the other will attach onto a microtubule from the opposite pole. Okay, right. And we're only showing 2 chromosomes here, but of course this would be true for the other 44 that would be in the human cell. Okay, now this is all occurring within the cytoplasm. So this next stage, after the nuclear envelope is broken down, is what's known as chromosomal capture, okay, in which the microtubules attach to the kinetochore of these sister chromatid pairs. Okay, right. Next up, what's going to happen is metaphase. And in metaphase, what happens is all of the sister chromatid pairs are lined up in between the 2 centrosomes. Now, the space in between 2 centrosomes, okay, or the line that is equidistant from the 2 centrosomes, is known as the spindle equator, or the mitotic spindle equator. So this line that is down the middle, which is an equidistance from the 2 centrosomes, this is known as the mitotic spindle equator. Okay, and these sister chromatid pairs are going to line up on the mitotic spindle equator. So I'll colour all of this in. So here is our 2 centrosomes here. Okay, and now we're going to get both of our sister chromatid pairs lining up like so. So here is our sister chromatid pairs of 2 red sister chromatids, and here is our sister chromatid pair containing the 2 blue sister chromatids here. Okay, and we also know, of course, that each of the 2 sister chromatids within a sister chromatid pair will be connected to opposite centrosomes, or to opposite poles of the mitotic spindle. Okay, like so. Right, so here are the microtubules that are attached to these kinetochores, like so. Okay, and that's metaphase now, and that's the final phase that the MCDK is responsible for. The next phase, anaphase, is going to be governed by a totally different protein, okay, one which is actively going to make sure that MCDK goes down. Okay, so there's now another very important checkpoint to cross, which is known as the spindle assembly checkpoint. So to move from metaphase of mitosis to the next phase of mitosis, which is known as anaphase, you have to cross this checkpoint, which is known as the spindle assembly checkpoint. For short, this is called the SAC. Okay, and once you've crossed this, what will happen is MCDK levels will fall very rapidly because cyclin B will be destroyed, basically, and a new protein will take charge of the entire orchestration of the cell cycle. Okay, or what's left of it anyway. And we'll see that in the next video.